Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is electron transport chain. The electron transport chain is a brief biochemical way of reporting describing a chemical reaction in which a donor molecule, a donor compound, transmits its a pair of electrons to an acceptor to an upholder in the presence of an enzyme. In simple words, we can also say that the electron transport chain is a set of enzymes which is present on the inner mitochondrial membrane, and it is the main source of energy generation in the aerobic pathway of energy production. Before watching the videos of electron transport chain, you also must watch the videos of Krebs cycle and glycolysis, and you must make their concepts clear about NADH, that is nicotinamide adenine dinucleotide, and FADH, that is also known as flavinadenine dinucleotide, that are produced during Krebs cycle and glycolysis. You must note that NADH is produced in both biochemical processes like Krebs cycle and glycolysis, whereas FADH is obtained only during Krebs cycle, not in glycolysis. You can find the link of these videos in the description of this video. Coming back to our topic, the main idea of electron transport chain is that the reducing equivalents such as NADH and FADH transfer their electrons via a series of electron carrier by oxidation reduction reactions to the final hydrogen or electron acceptor such as oxygen. So by the combination of hydrogen and oxygen, water is synthesized, water is formed during the electron transport chain. That is the main idea of electron transport chain. Now let's try to know about the electron transport chain in detail. The mitochondria has an outer mitochondrial membrane, inner mitochondrial membrane. A space is also present between the inner and outer mitochondrial membranes, which is known as intermembrane space. And inside the mitochondria there is present mitochondrial matrix. If we consider a closed section of the mitochondrial membrane structure, then we can visualize the structure of electron transport chain. So here we can imagine the outer mitochondrial membrane, whereas here we can imagine the inner mitochondrial membrane. The inner membrane space is present in its center. The inner mitochondrial membrane consists of four sets of enzyme complexes, which are named from enzyme complex 1, enzyme complex 2, enzyme complex 3, and enzyme complex 4. So these enzyme complexes are from 1 to 4 in Roman numerals. The mitochondrial matrix also consists of numerous other cycles such as Krebs cycle and beta oxidation. By watching these videos like Krebs cycle and beta oxidation, you will know that these cycles produce reducing equivalents like NADH and FADH. These reducing equivalents possesses electrons which are giving to this set of enzyme complexes. Electrons then travel from the first enzyme complex to the fourth enzyme complex. So finally, during this movement of electrons from first enzyme complex towards the fourth enzyme complex, energy is generated. This energy is utilized in pumping the hydrogen ions from the mitochondrial matrix into the intermembrane space. This continuous pumping of hydrogen ions into the intermembrane space causes the generation of higher concentration of hydrogen ions in the intermembrane space as compared to the matrix of the mitochondria. This produces a positive charge in the intermembrane space and the negative charge in the mitochondrial matrix, which is known as electrochemical gradient. The hydrogen ions cannot cross this electrochemical gradient because the inner mitochondrial membrane is non-permeable to hydrogen ions. So here we need a special transporter. The enzyme ATP synthase comes for proper functioning. The enzyme ATP synthase transports hydrogen ions into the mitochondrial matrix and utilizes the energy generated from the flow of hydrogen ions to phosphorylate adenosine diphosphate, shortly called ADP, to adenosine triphosphate, which is commonly known as ATP. This is the basic concept of energy generation in the cells in the form of ATP, which is also an energy currency of cell in an organismic body. Now let's study about the various electron transport chain enzyme complexes. What you can see here in the inner mitochondrial membrane, which has these electron transport chain complexes. These complexes are numbered from 1 to 4 in Roman numerals. In addition to all the 4 complexes, we also possesses 2 and other additional prosthetic groups called the Coenzyme Q and Cytochrome C. This one and this is also. So let's first talk about the complex number 1 of electron transport chain. Complex number 1 of electron transport chain is named as NADH dehydrogenase complex. It is also known as NADH oxidoreductase. This first complex is a L-shaped protein complex which is present in the inner mitochondrial membrane. This L-shaped protein complex consists of 2 arms. The first one is vertical arm and the second is horizontal arm. The vertical arm is present inside the inner mitochondrial membrane. On the other hand, the horizontal arm is present inside the matrix of the mitochondria. It is very easy to understand to consider the job of any enzyme which are functioning during the electron transport chain. So by just considering its name, its nomenclature, we can easily understand the function and job of any enzyme. So the enzyme complex 1, which is called the NADH dehydrogenase complex, it is so called because it is a dehydrogenase. Dehydrogenase meaning it removes hydrogen from its corresponding substrate. Therefore, it removes hydrogen from the reduced form of nicotineamide adenine dinucleotide, and it is called a complex because it also possesses flavinadenine mononucleotide and iron sulfur compounds. It is also known as oxidoreductase because dehydrogenation is an example of oxidation reduction reactions. The important and main function of this complex is to receive electrons from NADH and transport it further to the electron transport chain. Next, we will discuss about the complex number 2 of the electron transport chain. This second complex is known as succinate dehydrogenase complex. It is also called the dehydrogenase because it removes hydrogen from its substrate. In this case, succinate dehydrogenase enzyme complex, which is the enzyme complex second, will lose hydrogen from the succinate and oxidize it to the fumarate. This conversion of succinate to fumarate in the presence of enzyme succinate dehydrogenase complex is a biochemical step happens during the Krebs cycle. In this way, it is called a complex as it also contains iron sulfur clusters. In addition to succinate dehydrogenase complex, an additional and a second complex is also an integral part of the Krebs cycle, which chemically catalyzes the reaction for oxidation of succinate to fumarate. In this reaction, the reducing equivalent FADH2 is also produced, which is utilized for the transfer of electrons in the electron transport chain. The complex third of the electron transport chain is known as the cytochrome reductase. It is also called the Q cytochrome C oxidoreductase. All the cytochromes are a group of proteins which contain heme as their complexes or the prosthetic groups. A heme is a complex red organic pigment containing iron or other atoms to which oxygen binds. They also contain iron core in which the iron can exist in an oxidized and reduced form. Depending on the electrons, it has the complex. The complex third contains 3 types of cytochromes. The first one is cytochrome B, the second is cytochrome C1 and the third one is cytochrome C. The cytochrome C then transports its its electrons to the complex 4 of the electron transport chain. The complex 4 is called the cytochrome C oxidase. It is a heme and copper containing complex. The term heme I have already told to you. The main function of the complex 4 is to oxidize the cytochrome C, that is to receive electrons from the cytochrome C, and then it utilizes those electrons for the reduction of oxygen to water. In this way it is called cytochrome C oxidase. So students, all these are the 4 enzyme complexes of the electron transport chain. In addition to all these 4 enzyme complexes, another electron carrier which is known as the coenzyme Q10 or ubiquinone also performs function during the electron transport chain. Coenzyme Q10 is a ubiquinone that is chemically impart to vitamin K. Ubiquinone is such a stage of biochemical compounds that is found in all the living tissues that function as an electron transfer agent in the respiratory and some other tissues. Ubiquinone is any of several quinones that is found in most of living cells that function as a coenzyme which transfer electrons from one molecule to another molecule during cellular respiration. So coenzyme Q10 is a substance similar to a vitamin that is found in every cell of the body. So our body make coenzyme Q10 and our cell use it to produce energy according to our body needs for cellular growth and maintenance. It also function as an antioxidant that protects the body from damage caused by harmful molecules. Next, we will study the steps of electron transport chain. Now we try to consider the main steps of electron transport chain during which electron transport chain actually performs function. There is a clarification which sketch the membranous structure of the mitochondria as well as the organization of the different electron transport chain complexes. When we confront it with the structure of the mitochondria, we can visualize that it is the outer mitochondrial membrane that is represented here. On the inner side, here is present inner mitochondrial membrane. This inner mitochondrial membrane is represented here, which has shown by the space between the bottom two lines of the interpretation. The space between the inner and outer mitochondrial membrane is intermembrane space that is represented here, whereas the area, the space covered by the inner mitochondrial membrane is filled with a jelly-like material that is known as mitochondrial matrix. In this mitochondrial matrix, there are performed altered biochemical cycles like the Krebs cycle or the beta oxidation of fatty acids. These biochemical cycles prepare the reducing equivalents known as the NADH2 and FADH2. Now the electron transport chain activates when the NADH2 transfer its electrons to the first complex. In this first complex, the electrons are first received by the flavinadenine mononucleotide, shortly called FMN prosthetic group. The FMN then transfer these electrons to the iron sulfur protein. This movement happens by the redox reaction, during which the ferrous ions move from the ferrous state to the ferric state, depending upon when they accept and removed electrons. During the next step, the ferrous sulfur proteins transfer these electrons to the coenzyme Q. Q is the ubiquinol, which is a freely permeable molecule inside the inner mitochondrial membrane. These redox reactions, also known as oxidation reduction reactions, generate energy which is utilized to pump 4 hydrogen ions from the matrix of the mitochondria into the intermembrane space. The coenzyme Q then associates 2 hydrogen ions with 2 hydrogen ions from the matrix of the mitochondria and gets converted to the reduced form of ubiquinone. Now coming to the second complex, which is succinate dehydrogenase complex, which possesses the enzyme succinate dehydrogenase. It catalyzes the conversion of succinate to fumarate in the Krebs cycle. This is the oxidation reaction which creates the production of FADH2. FADH2 remain inside the second complex and contribute the electrons to the iron sulfur complexes of the second complex. Then these electrons are transferred to the ubiquinone in a similar way as these electrons are transferred from the first complex. The ubiquinone is a freely permeable molecule inside the inner mitochondrial membrane which is utilized to donate the electrons to the third complex. Here in this third complex, the electrons are first received by the cytochrome C1. The cytochrome C1 then donates the electrons to the iron sulfur protein which then hand out the electrons to the cytochrome B. All these reactions, all these transfer of electrons occur by the redox reactions that I have already explained in the first part of the electron transport chain video with more clarity. Then these electrons are transferred to the cytochrome C which is an intermediate protein utilized to hand out the electrons to the fourth complex. The redox reactions occurring inside the third complex creates the pumping of the 4 hydrogen ions to the intermembrane space. During the next step, the cytochrome C undonates the electrons to the fourth complex. In the fourth complex, the electrons are first accepted by the copper ions which are of A type. The copper ions then donate these electrons to the cytochrome A. The cytochrome A then transfer these electrons to the copper ions again which are of B type. These copper ions then transfer these electrons to the cytochrome A3. During the final step, the cytochrome A3 then hand out the electrons to the final electron acceptor which is the molecular oxygen. The molecular oxygen then combines with 2 protons from the mitochondrial matrix. These are the same protons that became separated from the electrons of the NADH2 and were dissolved in the mitochondrial matrix. This complete phenomena is also described in the first part. So due to this combination of protons, the molecular oxygen gets reduced and synthesizes water molecule. These redox reactions in the fourth complex causes the pumping of 2 hydrogen ions towards the intermembrane space. All this pumping of these hydrogen ions into the intermembrane space causes the generation of higher concentration of hydrogen ions into the intermembrane space as compared to the lower concentration of hydrogen ions inside the mitochondrial matrix. Inner mitochondrial membrane is an impermeable to hydrogen ions. So hydrogen ions cannot cross this impermeable membrane. As such, here comes the role of another enzyme known as ATP synthase. This ATP synthase is present inside the inner mitochondrial membrane and it utilizes the flow of hydrogen ions from higher concentration towards the lower concentration to produce enough amount of energy. This energy is utilized to phosphorylate adenosine diphosphate to adenosine triphosphate. This is the basis of ATP energy generation inside the any organism's body. The working of ATP synthase can be compared to the working of a hydroelectric power project. The water that is collected behind the dam can be compared to the hydrogen ions present in higher concentration in the intermembrane space. The turbines of the dam can be compared to the enzyme ATP synthase. When the water flows from the height which is compared to the flow of hydrogen ions from higher gradient to the lower gradient, and this causes the spinning of turbines inside the dam which can be compared to the functioning of ATP synthase enzyme. The power is generated inside the dam can be compared to the adenosine diphosphate that is produced to ATP synthesis. So this was the mechanism of the working of the electron transport chain. This is the electron transport chain in more detail and with the help of mitochondria and enzyme complexes because it is also an important topic that often comes during annual examination. So by understanding all of its steps well, you can write this chain in your own words like glycolysis and Krebs cycle. So keep on study your study very well and thanks for watching this your video.